Today, we'll speak about something that's a little bit significant. Every once in a while, there comes by a technology that changes everything. A revolution of sorts. This is a research I did. It's a review research. It doesn't produce anything new, it just collects what other scientists did. I don't have the courage to go with my colleagues, they're scientists. First, let me introduce the concept of technology. We all know what technology is, but can anyone define the word technology? Can anyone of you define the word technology? Technology is any way or tool that enhances the human life. So, here comes the question. What's the oldest, most significant technology that humans came up with? Any suggestions? The oldest technology that's very significant? Certainly not. Well, to my surprise, it was fire. But how can fire be that significant? We're in an age where fire is omnipresent. It's everywhere, and you don't even think about it. So let's look at an animal that has some similarities with us, but he doesn't know how to use fire. This species don't use fire. So, the gorilla, if we look at its skull, we'll see that most of the skull is teeth. Because the gorilla needs a lot of force to crush the wood that it eats. It needs a lot of energy and a lot of anatomical predisposition to be able to get energy out of its food. When we compare the human brain to the gorilla's brain, we'll find out that the difference is not just in size, it's in energy consumption. The gorilla needs to eat 60% of its time alive. It spends 60% of its day eating just to be able to support this brain. If we look at the human brain, we'll see that 2% of the human weight is actually the brain, but 20% of the human energy is spent from the brain. That's a very expensive brain you have. So, obviously fire gives us the ability to prepare food that's very calorie dense, that we don't need to spend most of our time eating. That's how brain development happens. We had time to communicate, and we had time to learn, to see, and that's how we developed this wonderful brain we all have. So, if we look at the human before fire and the human after fire, we'll see the changes are mostly in the skull. The skull became smaller in the jaw area and bigger in the brain area. And that was a breakthrough technology. The second breakthrough technology that happened was antibiotics and vaccination. And I'll not explain how this was very important and significant, but we can take a look at the lifespans of people. Before antibiotics, no one lived more than 100 years. It was a rarity, and when we look at the average was 31, and once we discovered vaccination, it jumped to 50, and after antibiotics, it jumped to now closer to 70 years old. This is a breakthrough technology. We changed our own selves through these technologies. Now, we're very lucky to be alive during the time that such a new technology, probably the most important of them all, is being developed. And this is how I introduce you to CRISPR gene editing technology. We all make choices, but then our choices make us. This can more be true than you can see in the genome editing. Genome editing is two things. You remove genes and you add new ones. Now, we all heard about gene therapy in the 70s. We all heard about genetic engineering. What's the difference? The difference is we before couldn't do it. We had dreams, but we didn't have the tool to be able to achieve it. Now, for the first time, we have a tool that's so precise and so selective that we can change a single base pair from a whole genome of an organism while it's still alive, while it's an adult. Before, you had to work with germ cells, work with zygotes, work with embryos. Now we can work with adult species. Now we're going with level samples. So this is Jennifer. Professor Jennifer is the one that discovered this system. The CRISPR system was simply known since the 70s, but they didn't know what it did. It was simply spacers. Spacers means chunk of DNA. They didn't know what this thing does. It's just there. And it was between ribosomal DNA. It was repetitive, and they didn't know what it is. Then they discovered that this is the defense mechanism of the bacteria against viruses. When a bacteria is attacked by a virus, it has only a few seconds to detect the genetic material of the virus and lesser time to destroy this material before it takes over the cell and turns it into a virus-producing machine. So what happens is, this enzyme, the Cas9 enzyme, along with genes from this CRISPR library that turned out to be a library of all viral infections that this cell and its ancestors had and survived. So they simply copy this gene, and they add here, it's called the guide RNA, and then it scans the whole cell, searching for any part, any genetic material that matches down to a single base pair, this guide RNA, and then it makes a double-stranded cut, which means that now we can remove genes and destroy genes down to a single base pair. That's a breakthrough. So how we can add genes now? We don't have to remove genes in double-stranded DNA. Now, how can we add our own genes? Well, it's simple. Cells have their own mechanisms of repair. They all repair themselves automatically. What we need to do is to provide our own genetic material in such high concentration that the statistics, the probability plays on our side, plays in our favor. When the cell attempts to repair itself, it uses our genetic material, it uses our sequence, our genes, and adds it to the genome. So, did it work before? Yes, it worked on mice, monkeys, human embryos, and HIV-infected cells. This was HIV DNA virus integrated into the human cell, dormant, waiting for the time to infect. And we were able to cut it out. And human neurons. So, the Fragile X syndrome is a syndrome known to cause the mental retardation in males, the most mental retardation caused in males, and it happens due to an defective gene. So what we did is that scientists actually were able to restore a fully grown neuron. It was a fully grown neuron with a defective gene. They isolated it from a human and inserted it into an animal, and they were able to restore it as if this gene from the beginning was sound. And it worked again. And it worked again. It worked again in human embryos in China that actually working with this technology, making changes to human embryos. So, now we're going into the concept of designer babies. We were thinking that now we can design our own embryos to the traits we want. Do you want to grow wings? We know how animals grow wings, okay? And the genes do you too. Do you want to have muscles? We know what are the genes that their overexpression produces muscles. Maybe we'll be able to cure the ultimate disease, which is death by disease or by aging. So maybe we'll be able to cure that. So finally, a fair warning though. When man blames God, men die. The last time someone attempted to do gene therapy, as my colleague explained, they died. But this was because they used the virus and the immune system actually had an anaphylactic shock because they used the virus as a victor. So I finally recommend that you watch shows that have the impact of these changes, like Altered Carbon. It's no show produced by Netflix, and that's it. Any questions?